I just want to welcome everybody to today's seminar. For those of you who don't know me, I'm John Rosine. I'm in the Exercise and Sport Performance Department, and it's a pleasure to have Dr. Diane Langford here. She's Professor of Neuroscience at Temple University in the Lewis Katz School of Medicine. Dr. Langford received her PhD from the University of Alabama in 1996 in cellular and molecular biology. She then went on to complete 5 years of postdoc training at the University of California in San Diego in the departments of medicine and pathology, where she trained in neuropathology and brain disease. After serving as an assistant professor at UCSD, she was recruited by Temple to the School of Medicine in 2007, where she began studies on brain injury. Her role as a professor of neuroscience, she studies a diverse array of central nervous system diseases that include HIV infection of the brain, substance abuse disorder, and repetitive mild traumatic brain injury. From her labs, they investigate changes in various clinical parameters, including blood biomarkers in athletes who sustain repeated subconcussive blows to the head. She is particularly interested in how the brain responds to injury and ways to promote recovery. She has received funding in excess of $10 million from the National Institutes of Health, the NCAA, and the Department of Defense and the Army Research Labs. So today she will be presenting on something that is near and dear to my heart in terms of concussive research, biomarkers in repeated subconcussion, and what do they tell us? And so please join me in welcoming Dr. Langford.
Hello. Please let me know if you can hear me in the back. Is everything good? Thank you. So this university is absolutely beautiful. I didn't really know what to expect, and I was pleasantly surprised. It's a beautiful campus, so welcoming. I'd first like to thank Dr. Ian Ming and Dr. Tamara King at the Center for Excellence in Neuroscience. Also, I'd like to thank Dr. David Moakler in the Department of Biosciences, College of Osteopathic Medicine, Dr. Karen Pardue at Westbrook College of Health Professions. And finally, I'd like to thank Dr. John Rosine, who's become a close collaborator in the past year or so. Dr. Rosine's work here at UNE is outstanding. And as I mentioned to many people yesterday when I was meeting everyone, this is really a gold mine for clinical samples in the athletes you guys have access to here. Your setup here is amazing. Many universities would kill to have this type of infrastructure that you are able to look at different athletes in many different sports at so many clinical parameters, including vision, balance, blood biomarkers, and so forth. So this is really a very rich environment to do these kind of studies. So what I'd like to talk to you about today is I'm going to start out talking about biomarkers in repeated subconcussion and how changes in these biomarkers might give us some information about changes in athletes' brains even before you know what's going on in a behavioral or a neurological sense. I'll also give you some, present you with some data that we've collected over the past few years. And then I'll finish with a little bit of in vitro work we do with human primary neurons in my lab, because at the end of the day, I'm a molecular biologist, not a clinician like Dr. Rosine and many of you are here. So this is hopefully very informal, and if I say something that doesn't make any sense, just please raise your hand and we'll clear it up before we move on. So first of all, I'd like to give you a really brief definition of traumatic brain injury. I'm sure all of you are familiar with this. It's the result of a direct hit to your head or your body where your brain shakes inside your skull. And depending on who you are and where you are in your life, things like the location of the hit to the head, the source of the force, the magnitude, and whether the direction of the force and the number of impacts you've sustained ahead of time and many other factors can really influence significantly the outcome. So you can have a very mild hit that will put you out for weeks, or you could sustain even a more forceful impact that you seem to recover from in a very in a rapid progression. So despite the clinical recovery, any brain changes that you see can result in the loss or dysfunction of neurons in your brain. And additional head injuries cause cumulative neuronal loss and dysfunction, and this is in large part responsible for cognitive impairments that you see. So what about the consequences of mild repetitive head trauma? So in a concussion, of course, one of the worst outcomes is coma, sensory and motor deficits. Also, epilepsy can be an outcome. Headache, sleep disturbance, which is becoming more and more important these days. They're finding more and more about the lymphatic system that is responsible not only for when you have a concussion, your sleep patterns are disrupted, but also when your sleep patterns are disrupted, you're no longer able to clear out things like hyperphosphorylated tau from your brain parenchyma. Memory problems, learning disabilities, and many affective disorders can be consequences of concussion. So from the American Academy of Neurology, I want to give you an update on a few of the risks and management of concussions. So individuals who are at increased risk for concussion based on scientific evidence, football, rugby, hockey, and soccer are at the highest risk. Females are at higher risk than males for many different poorly understood factors such as hormonal levels during the month. People say the neck musculature, the ball to head ratio. History of previous concussion puts you at risk for further concussions. Helmets help, but despite all of the claims out there, there's no proven evidence that mouth guards have any effect, that age is a huge factor. Position on the team, there's no proven studies on that. And, so there's many different things that underlie what causes poor outcome. A neurological exam following a concussion, of course, you check the eyes, vision, hearing, smell, motor tremors, head and neck injury, which are often overlooked, balance and neurocognitive functioning. I actually have an individual that works in my lab right now doing research on concussion that after he sustained a concussion, he could no longer smell. So it's just very strange how concussion affects different people. And I'll just give you a quick overview of some of the tests that you guys probably do all the time after a suspected concussion. You have the post-concussion symptom scale, standardized assessment of concussion, the SAC, and the balance error scoring, which is called the BESS. And the graded symptoms checklist is just how are your symptoms changing over time. And I don't expect you to read this, but it's just a measure of changing over time, which importantly must include baseline. Also, the SAC is a test that it's like a paper pencil test you can give for orientation, immediate memory, neurological changes, concentration, and delayed memory recall. The balance error scoring system, or BESS, is used, and when you're just standing on one foot with your hands on your hips, it looks pretty easy, but when you get on that little foam thing, as many of you know, it's a whole different story, even without a concussion. Imaging is a big effort focus right now because whether you get a CT or not, it's like putting the individual through the exposure to the imaging. Also, it's a time factor, and unfortunately, the fees associated with CT are also quite high, especially for individuals without good health insurance. So the FDA okays the first concussion blood test. So I'm sure all of you have heard about that, the GFAP, UCLH1. And basically this does not tell you if you have a concussion. What it tells you or not tells you, but what it suggests is whether or not you have a brain bleed and need a CT. So when they do this test in the emergency room, what they do is they take blood, they do an ELISA type analysis 3 or 4 hours later, which is too long to wait in most cases, according to the ER docs. Then they say, okay, should we do a CT or not? So it's really not the optimal thing right now. We cannot diagnose a concussion with a blood marker. Return to duty, play, or return to learn is different for every population, pediatric, adult soldiers, and even a professional versus an amateur athlete, not to even touch on the gender differences. The return to play field work or learn protocol is probably very familiar to you guys. It starts out with no activity, then you slowly introduce the athlete or soldier back into different activities related to their sport in particular until they can perform these things with no symptoms. So outcome post-concussion is very different, as I've mentioned. Sleep patterns, hydration levels, temperature, diet, medications, how much caffeine you drink, substance abuse, and of course genetics. We're doing a lot of work on genetics in my laboratory at Temple, and we do find that your genetic profile has a lot to do with your outcome post-concussion. So there are many different factors that you have to consider. So at the level of the cell in the brain, I'm sure you've all heard of diffuse axonal injury, and like other tissues in the body, when you have damage or pathology, it generally occurs where you have two types of tissue abutting one another. So in diffuse axonal injury, what happens is, for example, where you have the gray and white matter meeting in the brain, the tissue slides over each other, and it causes diffuse axonal injury where you can have the axons twisted, pulled, and stretched in a way that causes not only damage to the neurons, but disrupts all the cellular interactions in the brain, astrocytes, neurons, oligodendrocytes, and microglia, causes inflammation, and you have just the perfect storm. Even farther down, if you look at the organelle level, which is a big focus in my laboratory, is what you end up with is you have the mitochondria are very disrupted. The amount of ATP that astrocytes make after injury skyrockets. The amount of ATP neurons makes after injury plummets. So it's very dependent on the type of cell you're asking the questions about. So when you have an injury, your astrocytes make a ton of ATP, and they decrease the neurotrophic factors such as lactate or cholesterol that they normally provide to the neurons. So the neurons not only are in an energy deficit because they're making less ATP, they're also in an energy deficit because the astrocytes have just said, look, I'm not worried about you, I'm going to focus on myself for a while. So this is a very intense area of investigation in my laboratory. So now let's turn briefly to chronic traumatic encephalopathy that you've all heard about all over the news. And so what is chronic traumatic encephalopathy? Basically, what it is a collection of hyperphosphorylated tau in the depths of the gyri of your brain where the white and gray matter meet. So you can see here in the panel right here, where this little asterisk is, this is a collection of hyperphosphorylated tau, same thing down here. So as severity increases across the board, you can see accumulations of hyperphosphorylated tau quite robustly. So what happens at the cellular level is that, like your body, every tissue organ, you have a skeleton in your cells, obviously. In the neurons, you have a tubulin cytoskeleton that you see here. So what happens in chronic traumatic encephalopathy and other neurodegenerative diseases with tauopathy components, what happens is you have the stretching, tearing, twisting of the axons, and kind of, if you think about it, the ligaments or the tendons of the neuronal skeleton tend to become hyperphosphorylated, overly phosphorylated. That makes them not hold the two components of the tubulin together in the skeleton. So what they do is these little purple squiggles here, they let go, move away, clump together, and this allows the axon to unravel, which is never a good thing. So what leads to chronic traumatic encephalopathy? Studies now are saying that it's not a bad concussion, that it's repeated hits over time, whether that be concussion or subconcussion, which are milder forms of head trauma that are generally not even recognized and understudied. So what I want to move into now is to tell you a little bit about the studies we're doing at Temple. And what we do is we baseline every single NCAA athlete, every club, and every rec athlete. every year. So when they enter as freshmen or they sign up for a club sport or a rec sport, we bring them in and do a series of measures. And we've done over 3,000 so far in the past few years. And what it consists of is, importantly, the baseline, because you need to know where you're starting from before you can judge changes after a suspected head injury. So for this one, I want to describe one study to you that we did a couple of years ago, and it was done with the Temple football team. And like Dr. Rosine knows, you can't call up a football coach or some other coaches and say, hey, we want to take samples from all your players right before the game and then right after the game. I mean, they're like, what are you thinking? No, we're not doing that, especially if blood draws are involved, because a lot of people have their own ideas of how to get in the mindset and, you know, what if I give blood? How am I going to perform? So it's really difficult. And what makes this university unique is the support that you guys have from all your coaching staff with Dr. Rosine, where you can walk in and say, look, this is what we want to do. And also your athletes seem to be really on board with understanding what's going on in their bodies. So fitness and overall health seems to be a really key point in the population here at UNE. So what we did in the Temple football team is we took a series of measures. We do this every year during the August summer training camp, where we bring the guys in, we take blood, do vision, balance, the written tests, you know, like impact on the computer, impact testing, and some of the questionnaire type tests. And what we did is we did this every day there were, I think, 20, we started out with 30-something players and some of them injured out. So it was probably about 30 players where we took blood and did all the testing before practice and after practice many days in a row. So we had not only a lot of players that participated, but we had multiple measures on those players. So that kind of powered our study, if you will. Another important factor is some of the practices were no contact. Generally, those were held at the beginning of August, then followed by really heavy hitting for the remainder of the practices. And from what I hear, they hit really hard during practice. So to give you an outline of how this study looked, at the beginning of the study, we had 33 people. First day of training camp was no contact, then a series of full contact practices where we ended up with 29 players. So as I mentioned, before and after sports activity, which is practice in this study, we did the sport concussion assessment tool, the BESS, near-point convergence, and blood draws. We also asked them to wear an accelerometer embedded in a mouth guard. And the reason I like this accelerometer rather than the other kinds is this one is a boil and bite, just like a normal mouth guard. It clicks in your upper teeth here, so it moves with this part of your skull. Doesn't move with your hair, doesn't move with your skin like the patch, doesn't move with your helmet. It moves with your skull, and that's important. Even there's some mouth guards that fit in the bottom part, and that moves with your jaw, which is a whole different story. So that's why I like these mouth guards. Unfortunately, a lot of the players say they're not comfortable, I want my own mouth guard, So we have to respect that and use whatever product is out there that's the most practical for the sports team that you're looking at. Excuse me. So like all other accelerometers, the data is directly sent in live to our computer. A little avatar pops up and we know where the hit was, if it was linear, rotational, and if it was linear, which direction it went in. So this is a breakdown of the group characteristics of our players. I know you can't read this, but basically what it says is that they were matched for age, body mass index, number of previous concussions. We looked at the years playing football and position, and there were no significant differences across our players. So another factor I'd like to bring to your attention before we move on is we had players from high contact positions and players from low contact positions. And when we looked at the head kinematics, those two groups fell out in a natural grouping where there was a big range in the middle between those two. So the high impact groups had many more hits to the head, and the magnitude was significantly higher than the low impact group. So there was a big space in the middle where there was no overlap. And like I said, this fell out naturally. We had kickers, quarterbacks, etc. in the low impact group, and linemen, in the higher impact group. So that was what I think it was just lucky for us that we had such a distinction that we could have a natural separation. So first what we did is the scat before and after practice, and we also did near point of convergence. You can see here, there's many of you are familiar with this, I'm sure. It's just a ruler that you put right below your nose, and you move a little card closer and closer and closer. And the closer you get and can still read the letters on that card, the better your convergence is. So the farther away, it can indicate convergence insufficiency. So when your eyes cross or either the letters get blurry, then that's where we stop the measure. So we did this study as well, and this is an example of some of the head kinematics we took. So this is one hit. Peak linear acceleration on average was about 45, and you can see here, you can look at the linear from front to back, side to side, top to bottom, and the peak linear. So after all of the testing we did, when we asked them about their symptoms, they all felt great. Nobody had, no matter how many hits they got, no matter what the G-force, all the players, no, I'm good, I'm good. So that's why you see the flat lines down there from both the high impact and the low impact. So just asking a player is probably not the best. It depends on the player, obviously. But in this group, they all felt great. However, the near point convergence told us a different story. So what we saw in near point convergence is that the convergence score worsened over time in each individual in a very significant manner. So in our low impact group, the bottom line down here, there was really no change, no significant change. But in the high impact group that had, I think, 17 individuals, we had a very significant change, increase in convergence in that group that did not resolve until 3 weeks post-season when they rested. So what this tells us is basically this system is very susceptible to changes, and while they did recover post-season, what this is doing to the twisting and turning of the brainstem, which is the part of the brain that is responsible for maintaining a lot of the convergence, what is it doing over time? After you play 10 years, not only do you have normal aging, but you may have head hit related rapid or different aging patterns. So we see, for example, in individuals after they played contact sports for many years, they have a more rapid aging and up to 5 times increase in the risk for developing neurodegenerative diseases like Alzheimer's disease. So this is just a basic test that we did to see what was going on. So before we get into the blood biomarker studies, what I wanted to do is just remind you of the structure of the cells in your brain. So you can see the neuron here, and in the blue boxes you'll see some of the most popular biomarkers that people measure today. For example, the neuron enolase at the top and the UCHL1, so UCLHL1 is one of the ones that they're claiming is the concussion biomarker to tell if you need a CT or not. You also have others, the spectrin-dependent fragments that people at Penn and some researchers in Sweden are looking at. You have neurofilament light chain, neurofilament heavy chain. You have myelin basic protein, which is an oligodendrocyte myelin-specific one. And you have tau that we talked about a few minutes ago. You also have some glial cell markers that measure things like S100 beta, GFAP, and then you have some blood-brain barrier markers. So it's really important when you think about changes in blood biomarkers from the brain, you have to think about which cell this biomarker came from, is it brain-specific, and what it's telling you. So if you have an increase in something like hyperphosphorylated tau right after a hit, that tells you something very different than if you see a change in something like GFAP from an astrocyte. Because if you have an increase in hyperphosphorylated tau in your blood after a hit, that tells you your blood-brain barrier has opened up, right? And it tells you probably that if it's right after the hit, what's leaking into your blood or getting into your blood by a variety of mechanisms was probably there already, right? So if you see, for example, an increase in glial fibrillary acidic protein, GFAP, that just means your astrocytes got activated. It's kind of like an oh crap response, you know? The cells are like, oh my God, something just happened, and they increase this activation marker. So it tells you different. It tells you, are your neurons injured? Did they just get injured, or is this a thing that's been going on? And also, if you look at your glia, it's a response. And when you think back to the bioenergetics aspect of what I told you, the neurons are responding differently than the astrocytes. So do I think, or do you think, the more important question, there's going to be one biomarker that you can measure to say, oh, this person's had a head injury? Probably not. It depends on when you measure it after the head injury. It depends on the question you're asking. What type of cell are you asking about? So I think you want to look at things like GFAP, UCLH1, hyperphosphorylated tau, neurofilament light chain, and it tells you many different things over time about the change in the brain. And all of you, I'm sure, have heard of the secondary cascade signaling. So it's not just the hit where your neurons twist and your cells are like, oh my God, but after the fact, your brain starts to respond to the acute injury by changing all of its downstream signaling cascades that you guys are studying in the Neuroscience Center that are going to impact how you are able to respond. And changes in some of your genes make a difference in how you're able to mount your response to a head injury. So now I just want to give you a little bit of information about what we found in that football population when we looked at some of these most popular biomarkers. So S100 beta is one of the first ones we looked at, and I want to remind you that S100 beta is an astrocyte-enriched protein, and this one goes up when you exercise. So you can wind sprint, goes up. You can get hit on the head, goes up. So, and it's also not specific and only in the brain, it's in other parts of your body as well. So you have to be really careful with your interpretations of changes in S100 beta. So I'll show you what we found in our population. We also looked at neurofilament light chain, which seems to be a really promising marker. We looked at total tau because there's no good assay to look at hyperphosphorylated aberrant tau in the blood. And we also looked at one called pinch, which is one of my favorite proteins, and that's just like what I work on, so I'll tell you a little bit about that. So remember, we had two groups, the heavy hitters and the less heavy hitters, or the low impact group. So if you look at S100 beta in the football, you see you start out here, pre-practice, it's down. It goes up even after it was a no contact practice, goes back down, back up, back down, back up. So it's just up and down depending on what you've done. And interestingly, the heavy hitters and the low contact fell out in two different groups. So we saw significant changes, but you know, you have to be careful when you interpret this. When we did fancy statistics that I don't really understand, but my colleague who's a biostatistician helped us with, what we were able to do is show a trend whereby you could predict the change in S100 beta in nanograms per mil by the number of hits you got. So just for example, on this graph here, if someone got 10 hits, you expected the S100 beta to increase from 0.003 up to 0.025. So it fell out in modeling, but you have to, once again, as I mentioned, be cautious in interpreting these results. When we compared the changes in S100 beta on the x-axis to the changes in neurofilament light chain on the y-axis, we found that there was a significant association by these changes. But when we looked at total tau, and total tau, remember, is not just hyperphosphorylated aberrant tau, it's all the tau, the good tau, the bad tau, the ugly tau. So what we found is absolutely no association between total tau in the blood and the number of hits. And this was $10,000 later after paying a company, the Samoa company at Quanterix, to do these assays, because they have a platform that can measure the tiniest amount of tau in the blood by a super fancy ELISA assay, basically. So what we found is there was no association. So, you know, it's published, and it just shows you, even if your results don't show what you thought they would show or what you hoped they would show, you got to publish them anyway, because you have to publish all the data. You can't just cherry pick it. So now I want to move on to a new study we're doing with the Army, and for whatever reason, the Army is really interested in BMX dirt jumpers, right? So you can see this kid here. This is a kid who's 17, and he does this kind of stuff all the time, and then this happens. These are not the same people, so Bobby doesn't really fall as much as this guy over here. But at any rate, this is what we're measuring. And what we did is we took our team out to a place called Caddy Woods and another one called Posh, and these are out in the woods in Pennsylvania, and they attract professional BMX dirt bikers from all over the world. We got people from Japan, a lot of people from Japan coming here. So what we did is all the testing we did for the football team, before and after, we took blood, and what we're finding basically is that convergence is worse after, and there's some changes in blood biomarkers, as we saw in the football players, but basically this population, not all dirt bikers, but this population was a little older, and their lifestyles were quite different than the football players. So you have to take everything into consideration. So now I want to switch gears and talk about my favorite protein called pinch, and tell you how we're looking at changes in pinch in the brain and in cells from the brain to understand what happens at the cellular level. So this protein is produced during embryonic development to help cells migrate and polarize properly with each other. If you don't have pinch during development, you die. So after development and all the cells get oriented how they're supposed to in the brain, the expression of pinch goes way down. You can barely even detect it. But when you have diseases with a tauopathy component like epilepsy, Alzheimer's, HIV-associated aging, frontotemporal dementia, and chronic traumatic encephalopathy, pinch goes way up. So this is what it looks like. This is pinch. This is a little piece of the frontal cortex. You can see a little bit of the gyri here. And this brownish red color is pinch. So in a normal brain, someone who died with no CNS indications at all, you have no pinch produced. In a patient with HIV encephalitis, you see a huge amount, a very robust expression of pinch. And these are Western blots. How many of you have done Western blots? So as you know, you just take total protein from brain or spleen or kidney or cell lysate, and you separate it on a gel by size. So what we see here is that in normal brain, you have a little bit of pinch. In diseased brain, you have a lot. That corresponds with significant increases in hyperphosphorylated tau. This over here is an immunoprecipitation showing us that pinch and tau interact in the brain of an individual with a tauopathy. And this is another example of epilepsy. You have a lot. So these were actually resections from intractable epilepsy from my colleagues at USC in California. So this is epilepsy tissue, a lot of pinch, a lot of hyperphosphorylated tau. And this is what the tau looks like. Just to bring you back to the other slide, this is what tau looks like in the brains of individuals with CTE. So you see tau in the brown here accumulating around small vessels. You see the neurons here they're just gunked up with hyperphosphorylated tau, and a lot of it has left the cell bodies and found in the brain parenchyma. So this is what pinch looks like in chronic traumatic encephalopathy. You can see a small vessel here with very robust pinch staining. Here's a higher magnification image of a vessel with the red blood cells here and pinch around these vessels. Then when you do a double labeling in brain tissue from an individual that died with CTE, you see that pinch is in red, hpTau is in green, and where you see the yellow, that means pinch and tau are interacting. Maybe not directly, but they are expressed in the same areas around the same cells. So what does this mean? When you look at pinch and hyperphosphorylated tau in the cerebrospinal fluid of individuals with HIV, epilepsy, CTE, you see that they're both expressed in the CSF at the same time. But who's going to let you do that every day, right? Nope, not happening. I mean, the kids are already like, yeah, blood, I'm not so sure. But it is in CSF. Pinch is also robustly expressed in individuals in the CSF with TBI. So some of our pilot work, what we did is we did a series of soccer heading studies where we took soccer players, took blood, did vision, balance, blah, 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 like I did with a football team. And then we asked them to do 10 headers, just launching it from a jugs machine, 10 headers over and over and over. Then we took the blood, did the vision, balance, etc. And what we found is that pinch is increased in the blood after the headers at 1 hour and up to 24 hours in about 65% of the participants. So this is encouraging because we don't have to think about CSF, you can do blood. But still, a lot of people don't want to give blood before they do their sport or before they do their military activity. So what I've told you is pinch changes, patterns of pinch do change in the head and in the blood in the CSF of people who experience head impacts or have neurodegenerative diseases. So what my thing really is what happens in the cells. So I went into the laboratory and grew human primary neurons, and I thought about the secondary signaling cascade that happens after head injury. It happens when you have a neurodegenerative disease, and one of the main things that occurs is an increase in inflammatory factors like TNF-alpha. So basically what I did is I took some neurons in a dish and I squirted some TNF-alpha on there and I looked at them. The first thing we noticed, and we published this back in 2010 or something, is pinch goes up when you squirt TNF-alpha on neurons. Then we looked even closer and found out that the mitochondria in the neurons do something really funny. They leave over, you see at the control, all the green is mitochondria, and neurons have a heck of a lot of mitochondria. In a normal happy neuron, the mitochondria are in the cell body and all out in the processes here. But when you treat the cell, the neuron, with TNF-alpha, the mitochondria just scoot up around the nucleus. So, once again, energy deficit in the axons. So I'm going to end with this and just tell you what we've kind of drilled down to figure out what's happening. So in a normal axon, you have your mitochondria attached to your microtubules in the axons by all these proteins here, and you have the actin cytoskeleton also interacting with the microtubules. So this is a happy cell. This is what you want. Mitochondria move up and down, back and forth, the microtubules, to generate energy wherever you need it. But after an injury where you have TNF, you have a head impact, you have neurodegeneration, HIV, epilepsy, what happens is there's a huge calcium flux. This is caused by the secondary signaling cascade. This calcium flux, we've narrowed it down to show that it induces the promoter for the pinch gene. Pinch goes way up, goes over here, binds to actin in the axons, depolarizes it, and the mitochondria just kind of move away. So they go around the nucleus, so you have a big energy deficit in the neurons. So what I've told you is pinch goes up in neurons in diseases with a tauopathy component. Pinch interacts somehow with tau. It promotes signaling associated with increased tau phosphorylation. and it results in mitochondrial nuclear polarization. So overall, what I've told you is when you measure something in biomarkers, you have to ask all these questions. What cell are you looking at? What is the cellular response? Is it to the acute injury? Is it to changes in calcium? Is it to changes in things like inflammatory factors? And changes in these biomarkers are transient, and it happens over time. So you might have an acute response today, a different cell type responding tomorrow. So you've got to ask yourself, what question am I asking? So also likely there's a panel of biomarkers needed to get a clear picture of what's happening post-injury and in the outcome. So I hope I've shared with you a little bit about what we're doing at Temple and how we're looking at different aspects of head injury. And I just want to thank a lot of the people who worked on this project. As you can see, it's neuroscience in the School of Medicine, but it's also kinesiology, athletics, that's a key, engineering, ophthalmology, and some people at Hopkins and in Sweden. And this is just some people in my laboratory, so we have a great group, and this person in particular I want to point out to you. This is Leroy Wesley. When I did some of my football studies starting back in 2015, Leroy was on the team, so he was actually a participant. He gave his blood, he wore the mouth guard, and he, like many of you who are athletes and also in med school or in grad school, he wanted to know what happened. So after he graduated from Temple with a neuroscience degree, he called me up and said, hey, I want to get involved. So now he works in my lab, he runs the TBI studies, he's the phlebotomist, the measurer of everything, the analyst in the lab with all the ELISAs and so forth, and he's got that in with the athletes. He can go and talk to them on their level and also say, look, this is really important, da-da-da. So this is like a really full circle story. I think it's really cool that he moved all the way from one side of it to the other, and now he's pursuing a degree, a medical degree, and he wants to be a neurologist, like sports neurology type. So I'll leave you with that, and if you have any questions at all, I'd be glad to take them. Yes, sir.
Mine's a two-part question. The first part, have you considered using the impact test, or is that mostly just for concussions?
We actually do use the impact test. I'm sorry I didn't mention that. So that's part of our baseline for every athlete. Yeah, so we do use the impact test.
And then my follow-up to that is because I know with the impact in particular, but also I would imagine with the best and convergence, do you have any concerns about players altering their scores? Because I know like for NFL players, they've come out and said that they alter their scores. And I didn't know if you'd see that with CTE compared to concussions, where concussions you're determining whether they can go back into play. Yeah,
that's such a good point. And that your point was illustrated in the slide I showed you when we said, how do you feel? How's it going? Flat. I'm awesome. I'm great. Let's go back. So yeah, there are certain teams that are thought to do that more than others. And the best we can do is just hope for honesty. You know what I mean? And that's why we do multiple different tests that are not only how do you feel, but it's like the impact is kind of designed to like checks and balances or whatever. And then if we have a number, say, nope, your pinch is up, your tau's up, not going back. So that way we kind of take the pressure off the coaches, the physicians, and the players themselves, where they can't really just say, yeah, I think I'm good. So those are very good questions. Thank you. Yes.
Is it known at all what decreases pinch expression after embryogenesis? And then also, I think you mentioned pinch is implicated in migration and polarization. Mm-hmm. So when you have pinch overexpression, do you see any change in morphology or polarization in vitro or in vivo?
That's a very good question. And one thing I didn't tell you is pinch is also turned on in cells that have abnormal accumulation of misfolded proteins. And the cell itself is trying to survive. So in cancer, for example, pinch is also turned back on. And what we've figured out is happening is pinch is trying to promote cell survival even in the face of all these aberrant protein accumulations, and it tries to shuttle things like hyperphosphorylated tau to through the heat shock machinery and away from proteasomal degradation to kind of keep it alive. As far as why pinch goes down after embryogenesis, I don't know the exact answer to that. I can guess that it's probably once the cells are polarized and doing their own thing like they're supposed to do, it's just not needed as much until something bad happens. So when we overexpress pinch in the lab, the cells don't die. So they look pretty much the same, if you will. But what we have found is if we take cancer cells and prevent pinch from being overexpressed, like we knock it down with SH or siRNA, the cells become more susceptible to chemotherapeutic agents. So that's the best I know, yeah, for what might be going on. Yes, ma'am.
Sorry, just to follow up. So you don't see anything like axonal retraction or degeneration or anything?
When we overexpress pinch, yes, the axon, I mean, the cells are alive, but we do see process retraction when pinch is overexpressed. Yes, we do see that. Any other questions? Yes.
Can you go back to the S100 beta stuff? And you were talking about how you had your heavy hitters and your not so heavy hitters. And I know you said you had to be careful with your interpretation of what that rise in S100 beta really means. Did you consider at all like the exertional demands of the different positions? So I know you said like a quarterback or a kicker is in the light group, but the linemen are in the heavy group.
That's a great point. Yeah. And we did break it down by position, and we did not track exertion or hydration or have an activity monitor on them or anything. So that's one thing that when we published the paper, we had to include as a limitation, that we don't know if the players that had more head hits at higher magnitude were also the players that ran around the most. So that's a really good point. Yeah, that's exactly the type of thing that you have to be cautious about. Yeah. More questions? Well, thank you so much for your time, and it was a real pleasure visiting here at UNE. Thank you.